Welcome. Learn about mutation. In biology, a mutation is an alteration in the nucleotide sequence of the genome of an organism, virus, or extrachromosomal DNA. Mutations result from errors during DNA replication, mitosis, and meiosis, or other types of damage to DNA, which then may undergo error-prone repair, or cause an error during other forms of repair, or else may cause an error during replication. Mutations may also result from insertion or deletion of segments of DNA due to mobile genetic elements. A tulip flower exhibiting a partially yellow petal due to a mutation in its genes. Mutations may or may not produce discernible changes in the observable characteristics of an organism. Mutations play a part in both normal and abnormal biological processes, including evolution, cancer, and the development of the immune system, including junctional diversity. The genomes of RNA viruses are based on RNA rather than DNA. The RNA viral genome can be double-stranded or single-stranded. In some of these viruses, replication occurs quickly, and there are no mechanisms to check the genome for accuracy. This error-prone process often results in mutations. Mutation can result in many different types of change in sequences. Mutations in genes can either have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. Mutations can also occur in non-genic regions. A 2007 study on genetic variations between different species of Drosophila suggested that, if a mutation changes a protein produced by a gene, the result is likely to be harmful, with an estimated 70% of amino acid polymorphisms that have damaging effects, and the remainder being either neutral or marginally beneficial. Due to the damaging effects that mutations can have on genes, organisms have mechanisms such as DNA repair to prevent or correct mutations by reverting the mutated sequence back to its original state. Occasionally, mistakes occur during DNA replication and protein synthesis. Any alteration in the structure of a gene results in a mutation. Mutations occur during DNA replication when the chemical structure of genes undergoes random modifications. Once a change has occurred, the altered genes continue to replicate in their changed form unless another mutation occurs. Sometimes mutations occur during transcription or translation, causing protein synthesis to go awry. Although mutations may occur in any living cell, they are most important when they occur in gametes, because then the change affects the traits of following generations. Most mutations harm an organism. If a mutation occurs in a gene sequence that codes for a particular protein, the mutation may result in a change in the amino acid sequence directed by the gene. This change, in turn, may affect the function of the protein. The implications can be significant. The amino acid sequence distinguishing normal hemoglobin from the altered form of hemoglobin responsible for sickle cell anemia differs by only a single amino acid. Some mutations may be neutral or silent and do not affect the function of a protein. Occasionally, a mutation benefits an organism. Over the course of evolutionary time, however, mutations serve the crucial role of providing organisms with previously non-existent proteins. In this way, mutations are a driving force behind genetic diversity and the rise of new or more competitive species better able to adapt to changes, such as climate variations, depletion of food sources, or the emergence of new types of disease. Mutations can produce a change in any region of a DNA molecule. In a point mutation, for example, a single nucleotide replaces another nucleotide. Although a point mutation produces a small change to the DNA sequence, it may cause a change in the amino acid sequence, and thus the function of a protein. Far more serious are mutations that involve the addition or deletion of one or more bases from a DNA molecule. Adding or subtracting even a single base from a normal sequence during transcription can disrupt translation by shifting the reading frame of every subsequent codon. For example, an mRNA strand may include 2 codons in the following sequence, August UGA. The addition of a cytosine base at the beginning of the sequence shifts the spelling of these codons so that they read CAUGUG. This may result in an incorrect amino acid sequence during translation, or the protein may be truncated. Known as frameshift mutation, this type of alteration could result in the production of a protein with no real function or one with a harmful effect. Sometimes mutations are caused by transposition, in which long stretches of DNA move from one chromosome to another. These jumping genes, called transposons, can disrupt transcription and change the type of amino acids inserted into a protein. Transposons rearrange and interrupt genes in a way that generally improves the genetic variation of a species. While mutations can occur spontaneously, some can be caused by exposure to physical or chemical agents in the environment called mutagens. Common environmental mutagens include ultraviolet rays from the sun and various chemicals, such as asbestos, cigarette smoke, and nitrous acid. High-energy radiation, such as medical X-rays, can cause DNA strands to break, leading to the deletion of potentially important genetic information. Radiation damage can also affect an entire chromosome, disrupting the function of many genes. In chromosomal translocation, a piece of one chromosome breaks off and merges with another chromosome. In some cases, large sections of chromosomes may break off and be lost. The cell has highly effective self-repair mechanisms that can correct the harmful changes made by mutations and prevent some mutations from being passed on. Some 50 specialized enzymes locate different types of faulty sequences in the DNA and clip out those flaws. Another repair mechanism scans DNA after replication and marks mismatched base pairs for repair. Description: mutations can involve the duplication of large sections of DNA, usually through genetic recombination. These duplications are a major source of raw material for evolving new genes, with tens to hundreds of genes duplicated in animal genomes every million years. Most genes belong to larger gene families of shared ancestry, detectable by their sequence homology. Novel genes are produced by several methods, commonly through the duplication and mutation of an ancestral gene, or by recombining parts of different genes to form new combinations with new functions. Here, protein domains act as modules, each with a particular and independent function, that can be mixed together to produce genes encoding new proteins with novel properties. For example, the human eye uses 4 genes to make structures that sense light, 3 for cone cell or color vision, and 1 for rod cell or night vision. All 4 arose from a single ancestral gene. Another advantage of duplicating a gene is that this increases engineering redundancy. This allows one gene in the pair to acquire a new function, while the other copy performs the original function. Other types of mutation occasionally create new genes from previously non-coding DNA. Changes in chromosome number may involve even larger mutations, where segments of the DNA within chromosomes break and then rearrange. For example, in the hominin, 2 chromosomes fused to produce human chromosome 2. This fusion did not occur in the lineage of the other apes, and they retain these separate chromosomes. In evolution, the most important role of such chromosomal rearrangements may be to accelerate the divergence of a population into new species by making populations less likely to interbreed, thereby preserving genetic differences between these populations. Sequences of DNA that can move about the genome, such as transposons, make up a major fraction of the genetic material of plants and animals, and may have been important in the evolution of genomes. For example, more than 1 million copies of the ALU sequence are present in the human genome, and these sequences have now been recruited to perform functions such as regulating gene expression. Another effect of these mobile DNA sequences is that when they move within a genome, they can mutate or delete existing genes and thereby produce genetic diversity. Non-lethal mutations accumulate within the gene pool and increase the amount of genetic variation. The abundance of some genetic changes within the gene pool can be reduced by natural selection, while other more favorable mutations may accumulate and result in adaptive changes. For example, a butterfly may produce offspring with new mutations. The majority of these mutations will have no effect, but one might change the color of one of the butterfly's offspring, making it harder for predators to see. If this color change is advantageous, the chances of this butterfly surviving and producing its own offspring are a little better. And over time, the number of butterflies with this mutation may form a larger percentage of the population. Neutral mutations are defined as mutations whose effects do not influence the fitness of an individual. These can increase in frequency over time due to genetic drift. It is believed that the overwhelming majority of mutations have no significant effect on an organism's fitness. Also, DNA repair mechanisms are able to mend most changes before they become permanent mutations, and many organisms have mechanisms for eliminating otherwise permanently mutated somatic cells. Beneficial mutations can improve reproductive success. 4 classes of mutations are spontaneous mutations, mutations due to error-prone replication bypass of naturally occurring DNA damage. Errors introduced during DNA repair, and induced mutations caused by mutagens. Scientists may also deliberately introduce mutant sequences through DNA manipulation for the sake of scientific experimentation. Thank you for watching this video. Goodbye.